Hello, everyone. Welcome to tonight's event. My name is Tracy Johnson, and I'm the Dean of the Division of Life Sciences here at UCLA. And I just want to mention a few housekeeping items before we begin. This event is being recorded, so both the chat and the Q&A functions have been disabled. And due to our large number of attendees, which is very exciting, unfortunately, we won't be able to take your questions in real time. So thank you to all of you who submitted your questions in advance, and we're going to try to address as many of those as possible. So I'd now like to welcome you to our special virtual event, Novel Technologies for Alzheimer's and Dementia Treatments, featuring some of our remarkable faculty in the Molecular Biology Institute here at UCLA. But before I introduce you to today's moderator, Professor and Interim Director of the Molecular Biology Institute, Dr. Hilary Coller, please allow me the opportunity to tell you a little bit more about the Molecular Biology Institute's amazing faculty and students. The MBI, Molecular Biology Institute, is home to world-class faculty and students who are studying a wide spectrum of research areas that include, but it's not limited to, biochemistry, biophysics, structural biology, cell and developmental biology, gene regulation, immunity, microbes, and molecular pathogenesis. So the MBI was established in 1967 by the visionary Nobel Prize-winning scientist, Paul Boyer. And it was meant to serve as a hub for interactions between scientific groups on campus that was really focused on deciphering molecular structures and identifying regulatory crosstalk between living organisms. Since its inception just over 50 years ago, the MBI has been a catalyst for intellectual exchange across disciplines and has been able to transcend departmental barriers. So in fact, when I first came to UCLA some 8 years ago, the MBI was a major draw. And it was clear to me that the cross-disciplinary interactions within the MBI community would really take our research in new and unexpected directions. And this has absolutely been the case. So the MBI is really a stellar institute. And now I'm pleased to introduce you to its stellar interim director, Dr. Hilary Coller. So Dr. Coller received her undergraduate degree in biochemistry and molecular biology at Harvard University. And she received her Ph.D. in toxicology at MIT, where she studied the role of environmental chemicals as mutagenic agents in human tissue. The mission of the Coller Lab here at UCLA is to apply modern approaches drawn from molecular and cellular biology, genomics, computer science, systems biology, chemistry, and medicine, all to gain an insight into the molecular basis of a process called senescence, which is important and has important implications for health and disease, including cancer. Dr. Coller's research on the transition between quiescence and proliferation could have major implications for healing chronic wounds and developing methods to regenerate damaged and diseased tissues and organs. Many tissue-specific stem cells are, in fact, programmed to remain in a state of quiescence in order to avoid unnecessary cell division. So when injury is detected, these cells can enter into an active phase to produce cells that are needed to repair damage. If these cells are not able to re-enter the active state, full repair is not possible, which can contribute to chronic wounds and other disorders. This is just one example of many of the work in the Coller Lab that has far-reaching and important implications. Hilary has a history of just doing beautiful, beautiful research, and we're really thrilled to have her here tonight as our moderator for this evening's event. So please join me in welcoming Dr. Hilary Coller.
Good evening. Dean Johnson, thank you so much for that kind introduction. And thank you so much for your support for the Molecular Biology Institute. Thanks to all of you for joining us this evening. I have the distinct honor of leading the UCLA Molecular Biology Institute, and I'm really delighted to welcome you to the webinar this evening. The MBI facilitates research in modern biology by bringing together researchers with backgrounds in different aspects of molecular biology and helping them to work together to bring these different approaches to critical biological and medical problems. Today, you'll be hearing one really exciting example, which is the work of 3 different MBI faculty members who are all working on understanding Alzheimer's disease. Alzheimer's disease is a type of dementia that affects memory, thinking, and behavior. It's the most common cause of dementia, which is a general term for loss of memory. Alzheimer's disease occurs when memory loss is serious enough to interfere with daily life. And sadly, Alzheimer's disease is progressive in that it worsens with time, and we don't have cures at the present. So an estimated 6.2 million Americans aged 65 and older are living with Alzheimer's today. That is 1 in 9 people who are 65 or older has Alzheimer's dementia. It's more common in women, Black Americans, and Hispanic Americans. And sadly, by 2050, a projected 12.7 million people aged 65 and older are expected to have Alzheimer's. So Alzheimer's disease is characterized by an advancement through the brain. So at the beginning of Alzheimer's disease, there's often a loss of neurons, and this can usually affect the parts of the brain that are involved with learning. As it advances, the symptoms can become more severe, and it can result in loss of memory, disorientation, and confusion. There are 2 different types of molecular structures that are believed to underlie Alzheimer's disease. So one are plaques, and these plaques represent deposits of a protein fragment called beta amyloid, and it can build up in the spaces between the neurons. The other are these neurofilaments, or tangles, and they are twisted molecules that represent the protein tau that can build up within the nerve cells. And it's thought that the buildup of these aggregates can impede the communication between nerve cells and lead to their death. During the progression of Alzheimer's disease, both the plaques and the tau fragments, neurofilaments, will accumulate, and associated with this will be increased inflammation, a loss of neurons, and a loss of synapses. During the accumulation of these phosphorylated neurofilaments, what happens is that these tau filaments can be normally associated with these stabilized microtubules, which are kind of like the roadways of the cell. Once they become highly phosphorylated, they can leave the microtubules and clump together to form these neurofilament tangles. And it's been seen, and this is a recent paper in Science Translational Medicine from 2020, that the accumulation of these tau neurofilaments of neuronal tau. One of the reasons that the disease seems to be progressive is that these tau fibrils can build up within a cell, and then they can be passed to another cell. So it's thought that this might represent one of the ways in which Alzheimer's disease progresses over the course of time, is the spreading of these tau aggregates. So today we're going to be hearing from 3 fantastic MBI faculty who are going to be telling you about their research. We're first going to hear from Tamir Gonen. He's a professor who is developing new technologies for protein structure. We'll then hear from Professor Jose Rodriguez, who is uncovering the structure of proteins that contribute to Alzheimer's disease. And finally, we'll hear from Dr. David Eisenberg, who is going to be telling you about how we use the information about these protein structures to develop new Alzheimer's treatments. And with that, I'd like to introduce our first speaker, who is Professor Tamir Gonen. Professor Gonen is professor in biological chemistry and physiology. He's an HHMI investigator, and he's the founding director of the Micro-ED Imaging Center. Dr. Gonen's lab focuses on the structures of membrane proteins important in homeostasis and signaling. Dr. Gonen and his lab are pioneers in developing an exciting new tool in structural biology called Micro-ED that has revolutionized our ability to study membrane proteins at atomic resolution from vanishingly small crystals. Dr. Gonen earned a BS in inorganic chemistry and biological sciences and a doctorate of philosophy from the University of Auckland in New Zealand. He continued his postdoctoral studies at the Harvard Medical School. Please join me in welcoming Dr. Tamir Gonen.
Thank you for the kind introduction. Today I'm going to be telling you about a structural biology method that we've been developing in my lab for the past 10 years or so, and explain to you how we're using it to unravel mechanisms that lead to dementia and using that structural information to help design new pharmaceuticals and new treatments for dementia. Now, we're all structural biologists. The three speakers today, we're structural biologists, and structural biology is a branch of biochemistry that is concerned with how biological macromolecules are built. And that means when you have a protein, we're trying to figure out how they're built, and then based on the structure to try to figure out what they do, what goes wrong in disease, and then use that structural information to try to figure out what kind of drugs we could design to help those proteins along with performing their jobs. And structural biology has been incredibly important since the early days when Watson and Crick determined the structure of DNA, which helped explain to us how genetic information is inherited. Now, how does structural biology work? It starts with a crystal. Now, a crystal, you can think of a crystal as a three-dimensional object with repeat structural motifs inside. So here are two crystals. You probably recognize these. One of them is very useful, one of them not so much, and I'll leave it up to you to decide which is which. Both of these crystals are made out of exactly the same atom. This is just carbon. And you see that in this case, the arrangement of atoms is very different than the arrangement of atoms here. So that illustrates why it is important to understand the molecular structure of materials, because that explains to us something about the properties that we have to deal with. So you have a crystal and you shoot at it a coherent beam. In our case, it's electrons in an electron microscope, and that beam will get scattered, and it gets scattered in predictable ways. We record this scattering on a fast camera, and based on that scattering, which we also call a diffraction pattern, we can calculate back and say, what would have been the structure of this molecule here in the crystal that would give us a diffraction pattern that looks like what we recorded. Now, in my lab, as I mentioned, we've developed a method that's called microED, which stands for microcrystal electron diffraction. And it's a method that uses electrons as the coherent beam. And because electrons are so efficient with interacting with matter, we can get away with crystals that are invisible to the eye. So the crystals are so small, you don't even see them. They're about 1 billionth the size of what you need for other methods. Now, these are the kinds of electron microscopes that we need. They're incredibly impressive to look at. They're even more fun to use. This particular one is a microscope that we have at UCLA. We have a similar one at UCLA. It's not this exact one, but it's 14 feet tall. It's incredibly complex, and it has the ability to let us see atoms. The information, of course, is quite rich, and it needs very big computer clusters to do the image processing, and so we rely on heavy computations for our work as well. Now, how does microED work? We start with a small disk. It's about 3 millimeters in diameter, and inside we have those crystals that I told you about, and you see that they're moving because it's at room temperature. But now we're going to freeze them, and we freeze them because that stops the movement. It helps us focus on them and also protects them from radiation damage. We put the sample into the electron microscope, and so that is all done under cryogenic conditions, so very cold temperatures. We focus the electron beam on a crystal, and we start rotating the stage. So the stage is continuously rotating while we're collecting the data, and the data is recorded on a fast camera as a movie. And of course, at the end of that movie, what you end up with are many, many different frames, and each frame has one of those diffraction patterns that I told you about. And because we were rotating, we get all the different views of the molecule. This is one such example, and you see all these reflections, all these spots. We give them a position in X, Y, and Z, and then we extract from them the intensity, how strong that peak was, and that, of course, tells us something about the underlying structure. Because all this data came from a single crystal, we can fit everything together like a puzzle in 3 dimensions and figure out what the entire 3-dimensional pattern looks like. And based on this, we can then use the computational resources to try to figure out what the structure would have been to give rise to such a pattern. And in this case, I'm showing you a structure of an enzyme that we determined in the lab to atomic resolution by this method. Now, what I'm going to tell you today about is how we use microED to try to understand what happens in dementia. Now, there are some terms here that we need to become familiar with. One of them is that your brain is filled with receptors. Your brain is wired. It's wired to receive senses from the world. It senses your environment. And what does the sensing are these receptors. So when you eat something that is hot, it is a receptor that tells your body that the food is very spicy. If you touch something that is physically hot, again, it's those receptors that tell your hand to move back, move away, and be safe. Now, these wires are all interconnected, and when they start to fail in sending and receiving messages, that's when you end up with a condition that we know as dementia. And it's important to understand that dementia is not a single disease. It actually describes a combination of symptoms that together start affecting the quality of life that folks experience. And so this is just an example of the kinds of losses in sensing that happens in dementia. Now, your brain is wired through these long cells that are called neurons. There are thousands of them in your brain, and they're all connected with one another, and signals are sent and received. So in this simulation here, you can simulate this receptor in this area, and you see that the signal gets propagated and is sent throughout your brain to the correct place, so that you can then induce the correct response. Now, you may have seen pictures like these, where these connections are connections between neurons, which are called synapses, are illustrated like this cartoon over here. If you were actually looking at this in an electron microscope, this is what you might see. So here is one neuron, here is the opposing neuron, and right here is a synapse. They're making a synapse, and this is how they, this is where they communicate through. And if you were to blow this up and look at it closely in 3 dimensions, you would see that this is a membrane, and it's filled with these receptors that I was telling you about. So in my lab, we have a way of extracting these receptors. We induce them to form these crystals. So now you see these crystals, they're not diamonds, they're actually from these receptors. And you can use these receptor crystals in microED to obtain your diffraction data. And based on that diffraction data, you can then determine a structure. In this case, I'm just showing you an example of one of those receptors. And you see that on this side here of the receptor, there are these cholesterols bound, and there is a drug that we bound right here in the antagonist pocket. So this is an inhibitor. And once the drug binds here, that sends a message to the inside of the cell, where this part of the protein can then send the signals. And we're now using microED to solve and determine many different of these structures of these receptors with different drugs. And then the underlying goal is to identify and design new treatments for dementia based on a structural approach. And with that, I'd like to thank you for your attention.
Thank you so much, Dr. Gonen. It's really fascinating to hear about your amazing technological advances for determining protein structure. Next, I have the pleasure of introducing Dr. Jose Rodriguez. Dr. Rodriguez is an assistant professor in the Department of Chemistry and Biochemistry. His lab studies the complex architecture of biological systems, from single biomolecules to cellular assemblies at high resolution. His work combines computational, biochemical, and biophysical experiments. Dr. Rodriguez received a BS in biophysics and his Ph.D. in molecular biology at UCLA. Please join me in welcoming Dr. Jose Rodriguez.
Thank you. It's an incredible pleasure to be here with our remarkable panelists this evening, wonderful colleagues that are all part of the Molecular Biology Institute. I'm going to pick up where Dr. Gonen left off, talking about proteins that are incredibly important for the functions that we perform in our daily lives and essential for all parts of life. When proteins behave normally, they allow us to experience the world around us, to be human. But when they don't perform normally, we can encounter fundamental inabilities to perform essential tasks. And I'm going to talk about what happens when proteins don't perform normally, specifically a particular situation where proteins self-associate and form aggregates that are difficult for the body to deal with, and why that is associated with a condition like Alzheimer's disease. Now, we've heard about how Alzheimer's is a disease of the brain, and you can see indeed that in an Alzheimer's brain, there is a significant amount of damage compared to a normal brain. In fact, if you zoom in and take images of the types of neural networks in the brain that Professor Gonen was presenting in his presentation, you can see that in the Alzheimer's brain, you see an incredible number of deposits of proteins that are difficult for the body to clear. A large deposit is shown here, and it's difficult to make out the structure of the deposit itself. But if you were to disentangle the proteins to some extent in that deposit, what you find is that it's filled with these filamentous structures. These structures are not one protein, but many thousands of proteins that have all come together to form this one structure. They are found throughout the brain, but they appear progressively as the disease worsens. You see more of them spreading throughout the brain, ultimately affecting essentially all regions. Now, it's difficult to understand how it is that these molecules come together and associate, but it's helpful to think about it in terms of a simple analogy. The way we understand things as scientists is through observation and through relationship of our understanding of the molecules to our understanding of daily objects in the real world. And so when I think about molecules coming together, I think about a simple situation like a lock and a key. A lock and a key might come together to interact with each other, unlock the lock, and they even have a specific mechanism for doing so. But what happens if by some sort of aberration, you create or encounter a version of a lock and a key that are one, where the key is able to unlock itself? Then you can imagine these can come together almost ad infinitum. And so you can encounter a propagative event where the same molecule sticks to itself and creates many, many, many layers of this particular assembly. Now, in the very same way, two particular proteins happen to do the same thing in Alzheimer's disease. These two proteins are amyloid beta and tau. They're not, in fact, locks and keys. They're complicated molecular structures that are encoded by our own genome. They're proteins like the ones that Professor Gonen described, and they assemble with other copies of themselves to form these very long filaments. In fact, if we were to zoom in to one of these filaments, we can envision that there are many, many copies, identical copies of the same molecule stacked up through these horizontal layers that you see in this now atomic rendition of the structure found in these filaments. Each one of these layers is now a serpentine structure formed by the protein. Each one of these arrows in this magnified cartoon of a small region of this structure show you how complicated the serpentine behavior of the fold can be. And each one of the layers now represents a different protein adding to this assemble. Now, when we think about trying to tackle the problem of dealing with these assemblies, we have to attack it from multiple perspectives. My group and others in the MBI are thinking about these types of self-assembling molecules from the point of view of how it is that they come together, what it is that they look like at an atomic scale. And so we employ multiple tools to assess that. We employ biochemical characterization, biophysical characterization. We try in some cases to reconstitute the assemblies from scratch to see whether or not we truly do understand how it is that they come together. We also try to assess whether or not in a petri dish, for example, they might impact harm or induce some kind of pathology. We also can use animal models to try and test out whether or not the pathologies are also propagated in real life settings. And ultimately, we try to partner with those who can help us determine how to inhibit the types of pathologies that lead to disease. We rely heavily on the types of tools that Professor Gonen described, including electron microscopes, one of which you can see on the left-hand side of this slide, next to one of the staff scientists that we have that ensures that the microscopes operate properly. The samples that go into these machines are incredibly minuscule. In fact, a tiny drop of liquid can contain thousands of these filaments that can be found in the brains of Alzheimer's patients. You can image them directly. The filaments shown here are examples of these types of molecules. And if, again, we were to zoom in, we would see their atomic structure. But it's not so straightforward to actually witness that atomic structure. So in fact, to achieve this, we have to assemble teams like the members on this panel to try and actually reconstitute and sometimes visualize the exact arrangement of atoms present in these complex assemblies. So to do so, for example, we might take an assembly like this that represents this complex filament, and we might use the tools, the electron microscopes at UCLA, to actually interrogate its structure. In this case, I want to highlight one such structure, the structure of an amyloid-forming segment that is associated with Alzheimer's disease. This is the product of a collaboration, not only amongst the panelists that we have here tonight, but also other important members of the Molecular Biology Institute. And the structure that you see a chemical representation of here in the middle is actually modeled into the blue spherical objects that we see here, which are actual measurements, visualizations of the atoms inside of the assembly. This is from a side view. You can see the many layers of the assembly here. If you look at it from the top, you can see what a single layer looks like and where every single atom in this structure is. And that's the remarkable power of partnering together with the panelists to visualize these molecules. Again, this is a true team effort of many, many members of the Molecular Biology Institute, some of whom are highlighted here below. And it's a real pleasure to be part of this team.
Thank you so much, Dr. Rodriguez, for sharing with us your interesting research on the atomic structures of proteins that contribute to the development of Alzheimer's disease. Next, I'm excited to introduce Dr. David Eisenberg. Dr. Eisenberg is a professor of chemistry and biochemistry and biological chemistry and an investigator of the Howard Hughes Medical Institute. Dr. Eisenberg studies protein interactions with X-rays and electrons, bioinformatics, and biochemistry, with an emphasis on disease-related proteins. Professor Eisenberg's work has helped us to discover protein states that contribute to the development of Alzheimer's. He and his lab members are using this information to guide discovery of molecules that can diagnose and treat Alzheimer's and Parkinson's disease. Dr. Eisenberg received his undergraduate degree in biochemical sciences from Harvard College and his doctorate of philosophy and theoretical chemistry on a Rhodes Scholarship at Oxford University. Please join me in welcoming Dr. David Eisenberg.
Thank you, Hilary and Tracy, for making UCLA such a great place to make discoveries. And I really thank my collaborators, not only Jose and Tamir. We've had wonderful time, productive time working together, but others at UCLA. We are so fortunate in having basic sciences right next to our medical school. Makes a tremendous difference. And in our lab, here I list 16 of the current lab members who are staff scientists, graduate students, postdoctoral fellows who've worked on the projects that I'm going to briefly describe to you today. I'll be summarizing about 18 years of research in 8 minutes. So first of all, a little bit more about what is Alzheimer's disease and how it's related to these fibers of proteins. Then the question that undoubtedly you're thinking about, why hasn't our pharmaceutical industry been able to produce drugs and diagnostics for this disease? And if they haven't been able to, how can we possibly do it? I'm going to show you how. And the paths that we've taken are twofold. First of all, to discover through structure, through these structures that you've been hearing about, how to inhibit the formation of these fibers that cause Alzheimer's disease. And if the fibers are already there, how can we dissolve those fibers that have already formed? Hilary's given some background of Alzheimer's disease about how it starts in one region of the brain and then spreads to other regions. And if we take the brain of the autopsy brain of a person who died from Alzheimer's, we see cells, and we see inside the cells these so-called tangles, and those, if we enlarge them, are fibers of the protein tau. We also see these other aberrant structures called plaques of another protein, amyloid beta. Both these features were seen by Alzheimer's in Alzheimer, a pathologist in Germany in 1906. If we enlarge these tangles, as Jose showed us, they are fibers, and you get some idea of the size of these fibers. If about 10,000 are placed side by side, that would be the thickness of a human hair. So we have to use these tools of diffraction and electron microscopy to see them and to see where the atoms are, so we can do something about it. So why has pharma not come up with a drug yet? Well, a successful drug has to reach the right target and somehow inactivate it. And pharma, for historical reasons, has focused on not the right target. They focused on the amyloid beta structures, largely ignoring tau. And tau is the one, as Hilary showed us, which is correlated with the destruction of the brain. Pharma's also focused for drugs on antibodies. Well, they're very good at making antibodies. They're very useful drugs in cancer and other diseases. So they know how to make it, so let's try it for Alzheimer's. But they do not efficiently get into the brain, and they don't penetrate the cells in the brain where those tau fibers are. And at the basis of the problems that pharma's had, they've neglected to learn the atomic structures of these fibers. The atomic structures are so important if you're going to do drug design. You have to know where the atoms are. The reason they didn't do it is they thought it was impossible to do so. And I think it's our achievement at UCLA that working together, our team has learned atomic structures of tau, which is the cause of Alzheimer's, and alpha-synuclein, which is the cause of Parkinson's diseases. And having seen these structures, that allows us to design inhibitors of these fibers and other molecules which take them apart, that disaggregate them. So just to show you a detailed example, which came from the combined work of Tamir, Gonen, and Jose Rodriguez and our laboratory, here is a structure, and you see individual atoms here. So this enlargement to my screen is about 100 million times. And we see why these fibers are so stable. They're pairs of these sheets that bond together like a zipper. You can see the teeth of the zipper here. And most interactions between proteins, which are important in metabolism, they're like Velcro. They come together and they come apart. But this structure, it's like glue, like super glue. Once these fibers start to form in your brain, they're very hard to get rid of. So now I'm going to show you a little bit more in detail of how the fibers of tau, how they form. This is a segment of tau. It's the adhesive segment. And you can see the zipping together of the atoms in the tau fiber. And once this fiber starts, new molecules of tau add to it, elongating the fiber and causing problems and killing the cell. And the tau fibers are complicated. There's not only this one interface, but there are 3 different interfaces in which these fiber grows. And now we design an inhibitor. Here it's shown in red. and that prevents other layers of tau from adding to the fiber. You can see how frustrated the tau molecules are. They can't add to the fiber anymore once we've added our inhibitor. So that's how we stop fibers from growing. But what about fibers that have already grown? Well, recently we've learned how a small molecule breaks up these fibers of tau, and we're now designing drugs based on this knowledge. So here we see electron micrographs of fibers of tau, which have come, we've purified them from the brain, autopsy brains of Alzheimer's patients. These fibers all look the same, they're identical. You can see these bulges on them. If we add this molecule, EGCG, which comes, it's a dilute in green tea, but we add a lot more of it. And after 3 hours, it breaks up a lot of the fibers. If we leave it for 15 hours, it breaks up almost all the fibers. This is one we could see. You can see this is fibers going on the way to destruction after 15 hours. So because we know that EGCG breaks up the fibers, we determined a structure, which has just recently been published, of how EGCG binds to the fibers. So here I show you a magnified image of the tau fibers. You can see they're two J-like, J-shaped structures which come together at this interface. I'm only showing you 5 layers here. There are tens of thousands of layers in the Alzheimer's brain fibers. And I'm showing you in yellow where the EGCG molecule binds. And it binds like a wedge in between the place where the fibers meet. And that wedge drives apart the fibers and breaks up the layers. So we've learned how EGCG breaks up the fibers. But EGCG is not a good drug because it's metabolized into other compounds as soon as you swallow EGCG. But this knowledge allows us to design molecules that will do the same wedge effect, but can persist in blood and get into the brain and get into cells. And that's what we're doing right now. We've had some initial success with that, tested in the test tube and now being tested in mice, and we hope eventually in people. So just to show you a couple of the scientists who do this work, here's staff scientist Michael Sawaya, who's been working with us for 20 years, and graduate student Cindy Chang, busy at this business of determining structures. I know that you may have many questions. We don't have time to answer them all today, but here's my email. And if you send me email with other questions, I'd be delighted to answer them. So thanks for this opportunity to speak to you.
Thank you so much, Dr. Eisenberg. It's really inspiring to learn how the research in your laboratory is being translated into clinical studies. So now we're going to continue our conversation with all 3 of our faculty, and we're going to address as many of your questions as possible, and we appreciate those of you who have provided questions in advance. So the first question is, how does a family get their loved one involved in a study or new treatments? And for individuals who are interested in getting a loved one involved in a study, my suggestion is that you contact the Mary Easton Alzheimer's Center at UCLA because they are enrolling patients in studies, and they would be able to have a lot more information for you about the options for your loved one. So the next question is there a test to see if someone will develop Alzheimer's or dementia? And David, maybe you can get the ball rolling on this one.
Right. So that's an excellent question. And the answer is not yet, but there are many efforts underway. The most advanced method uses PET scanning. And it's been very good for showing the amount of amyloid beta in the brain. In fact, it's through those images that it's been discovered that amyloid beta does not in itself cause dementia, because with PET scanning, it's found that 1/3 of adults over 80 years of age who are perfectly normal they have normal cognition for their age group. Those people have brains just filled with amyloid beta, yet they're fine. But if you have tau aggregating in your brain, then you are demented. So that's been one of the powerful results of PET scans. PET scanning is also now being worked out for tau, and that will be extremely helpful. But there are problems with PET scanning. It's very radioactive, so you cannot have many PET scans. It's expensive. I think the reimbursement now is about $6,000 for a PET scan. And so it's not good for following progress of the disease or for testing drugs to see if they're working on a patient, because you can't have that many PET scans. So we need other methods. There are many laboratories hoping to find a liquid biomarker, that is, some compound in blood or maybe cerebral spinal fluid, which you can get through a spinal tap, which can tell whether a patient is coming down with Alzheimer's or Parkinson's disease. There's progress, but so far there is no reliable liquid biomarker available to us. That'll happen, I'm sure, in the next few years. In our lab, we're working on yet another method, and it's for MRI. And MRI does not use radioactivity. So patients can be followed and drugs can be tested. And it's about 1/10 the cost of PET scanning, and it's much more widely available. Most hospitals have MRI. So that's what we're doing in our lab.
Any other comments?
Well, I want to emphasize that the development of those probes for following the progress of disease is absolutely hinges on the understanding of how the molecules that track the fibrils interact with the fibrils. Getting structures of that is absolutely a high priority. And I think the work that we saw from David on the small molecules interacting with tau fibrils is just the beginning of that.
David, can I ask a follow-up question? So I was very interested in how you said that the amyloid beta doesn't track with actual dementia. What do you think the amyloid beta is doing there? Is it just a passenger? Or is it just a red herring?
The current hypothesis, Hilary, is that a buildup of amyloid beta in the brain can trigger the start of tau aggregation, but it doesn't necessarily do so. So there must be other factors.
And then my other question was, if you were able to reverse the tau aggregation, would that be sufficient? So could you get a neuron back to a healthy state, and would neurons regenerate to replace dead neurons?
Well, the body has amazing regenerative capacity, but I can't answer your question. What I think is our goal is to stop the progress of the disease. So as soon as it was diagnosed by one of the new methods or by a clinical study, patient losing his or her memory, and then started to take the treatment, at least I think we could stop the disease at that point. Whether there'd be regeneration or not, I don't know.
And we've learned from studies of other amyloid forming proteins, because amyloid beta and tau are not the only ones. In fact, alpha-synuclein was brought up, the cause of Parkinson's disease. There are many others. There's a prion protein that causes prion disease and so on. Many of these proteins have the same underlying mechanism of aggregation and cause of cell death and so on. Or it's hypothesized that they might have the same mechanism. And so you can see in other cases where animal models have been employed to try to understand the aggregation of these proteins, that if you halt the expression of one of these proteins after the aggregation process has begun, after disease symptoms are manifesting themselves, you can get an improvement just by halting the expression of these proteins or by clearing the proteins from the brain.
So I was wondering, the amazing wedging molecules that David was showing, could something like that be developed for alpha-synuclein?
Could it be what, Hilary?
Could one of those wedging molecules, could something like that be developed for alpha-synuclein also for Parkinson's disease?
I think possibly it could. And I think once one of these diseases falls through some sort of drug, the others are going to fall soon after.
That's an excellent point. Learning from how to tackle one type of protein aggregate is almost certainly going to teach us about how to do it for others. I think we need to find what those vulnerabilities are and then take that as a lead.
Another question I had in watching the videos was, how many molecules of tau are we talking about? Do you have a sense for that?
In a fiber?
Yeah.
In a single fiber, there might be there are tens of thousands of molecules. And as you see, there are many fibers in the brain. So many millions of molecules. Molecules are small.
And one of the challenges there is that your body continuously makes more. In fact, they're proteins that your body makes all the time. They happen to be entangled in this way. It's not what they're meant to do, but your body keeps making them because it thinks that that is a normal process. And so we need to combat that.
And it could be the origin of these diseases is there some breakdown in the regulation of how much the body is making, or a breakdown in the systems that we have at younger ages, which take care of the fibers and get rid of them. It could be that aging destroys the breakdown system as well.
And so one of the questions that we received is, what can one do to prevent or delay the onset of Alzheimer's or dementia?
Well, that's a very, very good question. It turns out that only about 5% of Alzheimer's patients have a direct genetic cause. That is, it's inherited. And most of those inherited Alzheimer's patients will know already because there are other family members, a lot of family members. Also, the genetically caused Alzheimer's disease starts earlier, probably before age 65. So that's just a small fraction. The other 95%, those cases are called sporadic, meaning that it comes from some environmental or personal cause. What could that be? It's clear that smoking is a factor in Alzheimer's, obesity, lack of exercise, disturbance of sleep. All of those probably contribute. Of course, they're correlations. They're not cause and effect. So it's very hard to know how much each of those matters. Lack of exercise is also correlated with the onset of Alzheimer's. What has made medicine a science is the double-blinded, placebo-controlled studies in which you have two groups, one of which is given the drug, one of which is given the placebo, and neither the patient nor the doctor knows which group is which. That's what's made medicine a science. Now, how do you do that with something like exercise? You can't have a group of people saying you can't exercise. So it's tough to identify those factors. But as soon as there are proposed drugs, then those double-blinded studies can detect whether they're effective. And you may have heard there was a news report in the last couple of weeks about a drug. I think it's donanemab from Biogen, which is said to have an effect on amyloid beta, and it was designed to diminish amyloid beta aggregation. And they said that it showed a small effect on patients. Well, I read the paper in the New England Journal of Medicine, and it's a very small effect. To differentiate the placebo group from the drug group is just a tiny difference. So my guess is that it may be approved, and I think that would be good. It'd be something, but it won't have a big effect, unfortunately.
So if I could ask a follow-up question, patients who inherit a susceptibility to dementia, or specifically Alzheimer's, what are the proteins that are most frequently associated there?
The most frequently associated proteins are either amyloid beta or its precursor protein. It's cleaved off from a larger protein, or the enzymes that do the cleaving.
I see.
Or you've probably heard another protein, ApoE. There are different types of ApoE. If you have type 4, you're more likely to get Alzheimer's disease.
Why is that?
I don't think it's known why.
Okay. So the next question that we received in advance was, what type of technological advances do you see in the next decade for this type of research? And Tamir, maybe you'd like to start the ball on this one.
Yeah, so over the last 10 years or so, the field of electron microscopy has really taken off. And so now we can investigate things that are much smaller and a lot easier to obtain. So you can, in the case of the samples that David and Jose, for example, are taking, you can extract those directly from brains and look at the morphologies directly from the cells. And so that is, I think, groundbreaking technology that's going to become very important in the next decade or so, simply because we weren't able to do these type of things before. And so as new technologies are developed, better cameras, more powerful microscopes, new lasers, new reporters that use fluorescence, for example, or UV to identify different things in cells directly, all of those are going to help with identifying these diseases.
And I have to say, the rate at which progress is being made now is remarkable. I think maybe David can comment more on this, but from the time that the first structure of an amyloid forming segment was determined to the time that we began collaborating all together with Tamir must have been, maybe 15 or so years, and a good handful of structures were determined. None from samples extracted from tissues, none from exact replicas of what you might find in the brain, all reconstituted in the laboratory in an attempt to mimic the ones that you might find in the brain. And in just the last couple of years, more structures have been determined by these methods than probably in many of the previous years combined.
And we're fortunate to having teamwork here, which makes a huge difference. And we have the pathologists who help us by giving us brain samples. And we have experts in the clinical aspects of these diseases in the medical school.
So our next question is also on the subject of guessing how long it will take for these treatments to become available to patients. And Jose, maybe you want to take a stab at it?
Of course, that's difficult to precisely pin down, but what's important is that we don't know when the next breakthrough is going to come. And so I think we've learned that in many other aspects of science. We're in the middle of a pandemic that we never anticipated would happen, and yet basic science has been able to pull us through every time because our investments in basic science pay off with remarkable dividends. And so I think what we need to keep doing is investigating finding possible avenues for treatment. And sooner rather than later, I think we will get there.
Maybe I could add that, you know, everybody is doing the best they can with the equipment that they have and with the technologies that they have. And then once in a while, there is a disruptive technology that arrives. And that makes things a lot easier and a lot faster. And then the floodgates open and then all these structures come in. And that's what Jose was alluding to just a few minutes ago. And so it's important to invest in those technologies. It's important to have that kind of support so that these disruptive technologies can be developed. And that would then help with the design of all these new drugs, because when we have those disruptive technologies, suddenly we see things in a new way, and suddenly we see things that we might have missed before. And that then clicks together and makes it possible to design new and innovative drugs.
Great. And then the last question for the evening has to do with collaborative research. So maybe you can talk a little bit about what it's like to be part of a team working on a problem.
Well, maybe I could start off with a story. In December 2013, I read Tamir's article in a journal. He was then working at Geneva Research Center in Virginia. And it was the first article describing his new method, microelectron diffraction. So I thought this would be good for the crystals that Jose and I were struggling with because they were so small. That method could work. So I emailed Tamir and I said, why don't we get together and work on this? I didn't hear anything back. So then I emailed him again. I didn't hear anything back. But that time it was after the holiday, and I phoned him, and then he said, okay, can one of you come here? So Jose went, and in no time mastered the method that Tamir had invented and determined that the first structure that I showed you. And then we thought it's important that we have Tamir at UCLA. So he came and joined us.
Yeah,
David, never let me forget that I did not answer the phone for the first couple of times.
I have to say it was a remarkable experience to be part of the team. In many ways, it was also, you know, a learning process. It's great to be exposed to new technologies. It's amazing to be able to use them for such important research. And I hope that in the years to come, there are many, many more such partnerships that lead to new innovations and new discoveries.
Terrific. Well, thanks so much to the three of you. Thank you to everyone for joining us this evening for this webinar on novel technologies for Alzheimer's and dementia treatments. We really hope you enjoyed the presentation and the conversation. This webinar has been recorded. We will send a post-event email with the link to the recording. Please feel free to share it with others. Thank you again for joining us tonight, and have a good evening.